Can you imagine living for centuries cancer-free? The bowhead whale seems to have evolved a biological escape route. Dr. Denis Firsanov and his team have unlocked one of the critical secrets to the puzzle of genetic stability and extreme lifespan. Join us today as we unlock the mystery that has enabled the bowhead whale to become the longest lived mammal on Earth. Could this be the next chapter in the discoveries on longevity and resilience? Jump into the conversation with me and let's explore the tantalizing possibility of whether this could unlock the future therapeutic target for human lifespan extension. Thanks for listening to the Saving Sapiens podcast. I am so excited to welcome Dr. Denis Firsanov, the lead author of a brand new paper on bowhead whale longevity. The bowhead whale is a famous and fascinatingly long-lived mammal species and represents one of the most interesting evolutionary niches. Dr. Firsanov, welcome to the podcast.
Thank you, Matt. I'm honored to be here.
So the average whale usually lives somewhere in the ballpark of a human lifespan. How long do bowhead whales live?
I think for now, the maximum estimated lifespan for bowhead whale is 211 years. And they were found already several bowhead whales who outlived 200 years. So I think we can be more or less confident in that. Definitely almost twice as much as human lifespan.
Now, the way that a species becomes long-lived has represented one of the most interesting and mysterious facts of biology. For those in our audience who are unfamiliar with Peto's paradox, can you talk to us about just why it should be so difficult to make an organism with this many cells actually live this long?
So it's, I think, now already established that cancer does not happen spontaneously, momentarily, and requires certain steps, so accumulation of mutations. And the more cells divide, the more mutations they should accumulate, or may accumulate just by chance or probability. And we can imagine that big species, like large body size, they have more cells in their organs. So each cell is supposed to divide more. So theoretically, the chance of accumulating mutations should be higher in large body size animals. And because of that, theoretically, they should develop cancer more frequently. But that's actually not observed in nature, and that's what's called Peto's paradox. So larger species do not have high incidence of cancer. That's what we can say. And that is why for us as humans, it is interesting to figure out why. What make each long-lived large species be resistant from cancer.
So it's a little bit like a lottery ticket, only in the wrong direction, right? It's the lottery nobody wants to win. If you're a species with very few cells, you don't have a lot of lottery tickets. You have kind of a naturally low chance of winning the cancer lottery. But when you are a large species, the way you acquire such large size is to have more and more, trillions more cells. And therefore, you have trillions more of these sort of cancer lottery tickets to draw. How has the set of evolutionary adaptations in bowhead whales protected them from winning the cancer lottery? And did it follow the expectations based on what we know from other long-lived large species like elephants?
So with elephants, it was shown that they have multiple copies of the tumor suppressor p53, and basically, so when something wrong with the cell does not have that many options. Uh, Elephant will evolve in the way that if something is wrong, it's better to kill the cell. And that's what multiple copies of p53 or high p53 activity does. It forces, if something is wrong, cells just undergoing apoptosis, so programmed cell death. And it's like a no cell, no bad cell, no future problem. That's how elephant evolved. And of course, we initially suspected that maybe bowhead whale have the same mechanism. So we applied different genotoxic stressors to the bowhead whale cells and surprisingly observed that they do not die more efficiently than human cells or other species. And we even checked the p53 activity in bowhead whale cells. But it would be too simple to be true, and we were excited that from one hand it's not the same mechanism, which make whale more interesting for us. So whale cells do not have p53 activity higher or more efficient apoptosis events. Another thing which we can think about, so somehow if the cell is damaged, so it can either die or let go to the permanent cell cycle arrest, cellular senescence. This is also a very powerful anti-cancer mechanism and also can be regulated for p53. And we also did not observe differences from human cells. So as human cells, they undergo cellular senescence the same efficiency. So it puzzled us a little bit. But another, the third option, I'm beneficial for the cell, this would be to avoid the situation when accumulating of mutations of genomic instability. That's what we found apparently bowhead whale does. So it avoids the situation when cells are accumulating mutations, so genomic instability, by efficiently repair all DNA damages. So from the evolutionary perspective, it made bowhead whale very unique. So how it found its niche to live so long, be big. I think it's the second largest animal on Earth, I mean mammal on Earth, which has so many cells and yet does not have cancer or age-related diseases.
That is an absolutely fascinating reminder of the fact that the adaptations are never goal oriented. Each clade of organisms has to essentially deal with the randomly dealt hand of evolution. And in the case of elephants, the expanded copy number of P53 gives it this aggressive apoptotic control system. But in the case of whales, because they lack this expanded copy number of P53 in the way that elephants have, they have this entirely alternative system. Can you speak a little bit more on the specifics? Because you guys actually were able to drill down how it does such an effective job at repairing DNA and keeping cell lines healthy in this alternative path rather than simply increasing programmed cell death.
Sure. I think I also probably want to comment on this, like expanding p53 copies as a programmed cell death mechanism. So you can think that it is actually good, right? But you need to have some other adaptations for that. So you can imagine if you have like inside your body damaged cells and they just die, so they have to be substituted for something. Or cellular senescence, if they stop dividing and stop functioning as what they're supposed to, they also need somehow to be eliminated and substituted. So we can like imagine that elephant might have like an expansion of stem cells, which can repopulate those removed cell population. So in humans, or like in mouse, they would made like P53 all they express in mice. Uh, and yeah, they have less incidence of cancer, true, but they still live short because they develop age-related diseases faster.
Yep.
So there is a balance. You cannot like so the strategy what Bowhead whale does, it keeps the same cell there apparently by repairing it. So there are, as you know, every day in our body we experience DNA damages, all variety of them due to natural metabolism or exogenous sources of radiation or something. And we checked pretty much all DNA repair pathways, the major one in Bowhead whales, their activities. And one specific, which considered to be the most dangerous type of DNA damage, DNA double-strand breaks, because it is well established that if unrepaired, DNA double-strand break can cause cancerogenesis and genomic instability. So we found that in Bowhead whale, double-strand break repair pathway, both of them non-homologous and joining and homologous recombination, are more efficient and uh, also more accurate because non-homologous and joining is major pathway in all mammals, in us also, because it works in all stages of the cell cycle, but inherently it's error prone. Basically, we evolved in a way that if we experience double strand break, then it's beneficial to fix it somehow, then leave unrepaired. Even if it causes deletions or insertions, which can disrupt like genes. We're lucky that 90% of our genome is non-coding. So if deletions are happening there, likely it won't cause immediate consequence unless it happened in the gene
I hope the audience registers that, by the way,
region.
because there's this long term argument over the C value paradox and why we have these profoundly inefficiently packed information containing units in the genome. Why is it that only 2 or 3% of the genome codes for proteins? And why is it that we have all of this allowed transposition and replication of all these transposable elements that occupy like half the genome, and why 90+% of the genome doesn't code? But you hit on something incredibly important, which is that because we're not single cell organisms, we actually benefit in somatic cell replication from the low information density of the genome, because when you have these breaks, there's like a 90% chance that they're going to occur in a non-coding region. That's a fascinating trade-off to the inefficiency and the bulk of the genome.
Right, right. But now those non-coding right, regions, they're very extensively studied. And also in our lab, there are groups which are studying line one transposable elements. Uh, they play very big role in aging and regulation of other genes. So those which people consider junk regions of the genome, those 90% or something, it's, I think, no more we can say junk. It plays an important role. It codes like non-coding RNAs, which plays a role in regulation of gene expression. Uh, it codes transposable elements, which can be bad or good depending on where they are and what they do. So even though the chance that double strand break will occur in non-coding region is obviously much higher, uh, but it still have like a possibility that produce large deletions, which will cause maybe the deletion of a fragment of the coding gene. happen in the close proximity of it. But in Bowhead, where what we found fascinating that their non-homologous end joining is also more accurate than in humans or in other species. Although most reviewers cared about humans, we had like mouse and cow. No one cared about those. Everyone cared about like how it's different from human. So when we sequence, so we had like special assay how we measure the activity of double-strand break repair, and then we can induce the break, allow cell to repair it, then isolate DNA from there and sequence the region around known place of double-strand break, and then see what happened there. And we found that in Bowhead there are much less frequency of deletions than human cells have. In human, it's well known that this pathway causes frequently deletions. And bowhead whale, so bowhead whale have not only more efficient DNA double-strand break repair, but also more accurate. This prevents significantly potential genomic instability in bowhead whale. So whale cells somehow evolved to have these very accurate and efficient repair pathways to deal with. So to avoid that situation when mutation even accumulate in a whale genome, so it does not come to the point that it reaches those 4 hits which are required for whale cells transformation.
So this prevents the dependence of whale cells on responding to multiple hits because of the fact that they're higher accuracy. Can you explain to the audience how you tested, first off, what the multi-hit hypothesis is in cancer, and then how you tested specifically whale cells to see if they were tolerant of more versus fewer hits?
Sure. So multi-stage hypothesis persists that cells require several mutations to become fully transformed. And it was shown that, for example, for mouse, to transform primary mouse cells, it requires just 2 hits. Mouse cells already have telomerase activity, so it's constantly expressed, and telomerase is active in somatic cells, which is not true for human, for example. All our cells do not all, like we have telomerase activity in stem cells and in germline, but in most somatic cells, we don't have it. Then mouse cells require to inactivation of one of the tumor suppressors, either p53, the most famous one, or retinoblastoma, RB tumor suppressor. And on top of that, you want to add oncogene. It's RAS oncogene. There is the way how you can test it, you can integrate oncogene, which has a mutation RAS V12G. So this RAS protein becoming very active and does not care about stimulus. It's always active. So this is sufficient enough to transform mouse cells. For human cells, humans are evolved, we are much more protected from this event. We require 5 hits. Telomerase, oncogene RAS, inactivation of 3 tumor suppressors, p53, RB, and p10 or pp2a. These are the p10 and pp2a from the same pathway. These are phosphatase. So once you have all 5 hits, human cells becoming transformed. So how you can test it?
And by the way,
Basically,
for the audience, when we talk about the cell line being transformed, we're specifically referring to the fact that they are essentially exiting normal somatic lineage to enter a cancer cycle.
um, so it is not like that simple to observe in a cell culture like that. If you just culture transformed cells or primary cells. They both will divide, but and maybe transformed cells will divide faster. But how you test it, there are two ways. First simple way is so-called soft agar assay. When you put agar layer on the bottom of a petri dish, and then you mix your cells with another layer of soft agar or agarose and put it on top. Primary cells, they require substrate, so the surface to grow. Transformed cells, they don't care about this. So transformed cells will grow without the substrate, so they will form colonies in agar, and only transformed cells do. So by doing this simple assay, you can see if cells form colonies in soft agar, then they are transformed. The more advanced, and the better way is when you inject those cells into the nude mice, so immunodeficient mice under the skin. And then you observe, so if cells are transformed, they will form tumor on mice. And this is an indication of the cellular transformation. So these are two assays which we use to check how many hits are required. So for Bowhead whale, we found, initially we hypothesized that if to follow this logic, mouse require 2 hits, human 5, but maybe whales that require 6 or even more to protect themselves. But we were able to transform whale cells just with 4 hits, inactivating p53, RB, adding telomerase, and oncogene RAS. So no fifth hit was required to fully transform whale cells. And it was very puzzling situation because that's where we started. That's where we started to see like what makes the whale cells be cancer free. And we found it's 4 hits that wow. So they can be easier transformed. And as we discussed, they do not undergo apoptosis more efficient. They senesce the same efficient. What's going on with those cells? Why do
for the folks that haven't read the paper, go read the paper. You're going, wait, how do they live so long? There's a lot of good papers and some speculative papers in the aging journals. Your paper is so great because it reads like you're walking through a mystery novel. You're like, huh, okay, it's fewer hits to transform a whale cell than a human cell, and they senesce at the same rate. What is going on? So lead us down the mystery novel. Where did you go next?
Yeah, exactly. So that's why like we hypothesize the way. So if they require less hits and don't die or senesce if DNA damage occur, so they might somehow avoid that situation of accumulation. So then we checked if they're more resistant to accumulation of mutations of various stressors, or our collaborators also sequenced those tumors from transformed cells. We found that they form less structural variants and mutations, so they were somehow inherently protected from accumulation of those mutations. And that's where, yeah, then it must be DNA repair. What else deals with the mutations and prevents them from accumulation?
So after you guys find this enhanced DNA repair mechanism, tell us a little bit more about what you did to sort of verify its activity and test its activity in other cell lines.
So you mean how we found what is responsible for that high activity, right? So you know, uh, there are many known DNA repair proteins. We did proteomics, transcriptomics. I did a bunch of Western blots because the reason why it is cool to study mammals, different mammals, not model organisms like mice, because we technically have the same genes. They just can be variation in the nucleotide sequence, like amino acids, uh, or some regulatory elements, but they're essentially the same genes. So by checking what is different, we can speculate how the species is different this or that pathway. So there was actually very uh, what pays our attention to this cold-inducible RNA binding protein, which we found to be very abundant in a whale. Long time ago, uh, me and my student, we did experiments putting bowhead whale cells into the cold room. It's +4 Celsius uh, for a week, and together with human cells. And surprisingly, we saw that whale cells uh, survive much better in a cold environment. Even though whales are mammals, right, and even they live in Arctic Sea, which is very cold, they have like a huge layer of fat, which like maintains the body temperature. But some of this can be exposed to the Arctic temperature. So whales are very resistant. And we knew that there are two cold shock proteins known in humans or like in mammals, cold-inducible RNA binding protein, CIRBP, and RBM3. And we checked the expression on Western blot simply. And that we found that CIRB is so much abundant in whale cells comparing to human. Then we looked at our proteomics and transcriptomics data. Said, hey, it's there also, very high abundant, and they live in cold water. Cold-inducible protein is hard. It was shown that that protein, it was a paper in PNAS, showed that this protein plays a role in double-strand break repair, I believe in 2019. And then we're going to think about, yeah, that might be it. So even though there are many other proteins differentially expressed in bowhead whale, like we had a bunch of lists, which we listed, I think, in extended data figures in our paper. But that protein really stands far, especially it's very impressive on Western blot. So then we have to say, well, if this is true, then if I make this protein be a lot in human cells, then it should improve DNA repair there. And that's what we did. We cloned that bowhead whale gene into the simple vector, transfected it into human cells, induced double-strand break there, and found that, yeah, it improves DNA double-strand break repair efficiency in human cells. This was a principle, but not enough. Okay, if it is important for whale, what if we knock down it, so make it less in whale? And it decreases the same pathway efficiency. So that's what make I find it fascinating, that we found the gene which is so much in the whale, and it plays role in all those processes which we found, phenotypes which we found before. So I would say, because nowadays, omics is very popular, but sometimes to make a discovery, you need to make an experiment which may sound stupid, like putting cells in a cold room, because CIRBP is not from the canonical DNA repair pathway. I would say it's not well established. Like in DNA ribosome break repair field, there are Q70, Q80, DNA PK, ATM, MRE11, RAT proteins. all core proteins are already established. People are now trying to find cofactors which help for this or that pathway. But CIRBP is not well characterized and established. So that long time ago, I think cold experiment helped us to focus on that particular protein
One of
source.
the common instant reactions by a lot of the community interested in extending human lifespan is to read papers, to follow projects like yours in a particular species that's being studied, and then to immediately jump on this idea that this is going to be the mechanism that explains this either longevity in a particular cell line or lifespan in a particular species, and then to immediately extrapolate that perhaps we could use this newfound secret of the biology of this one long-lived species and move it over to humans. Can you talk a little bit about sort of how this doesn't necessarily reflect transferability across other species, but how it reflects the environment in which this species actually evolved? And you kind of got into that already with regard to the cold adaptations.
Sure. I think for me, the goal for so you mentioned the extending lifespan, right? I know there are many scientists who believe that we can significantly extend human lifespan, but I would say that I believe more in a health span because I think like the health span, so we can delay the aging. And aging is, as you know, is a multifactorial process. There are too many things, too many hallmarks. And genomic instability is one of the hallmarks. So I know that people like usually when they want a magic pill, right, which they can take and live longer. But I think it's a combination of the process, like calorie restriction, intermittent fasting. I think it's already more or less established thing, interventions which we can prolong your health span and delay accumulation of age-related diseases. What we eat, how often we eat, this all plays the role. And our lifestyle, if we go to the gym or simply sit at home and do nothing. So these are very important things which people should consider. I don't believe there would ever be a pill which you go to McDonald's or other fast food, drink alcohol like crazy, smoke cigarettes, and take a pill and nothing happen. I mean, it's not going to work that way. With our finding for the bowhead whale, this, from its name, cold-inducible RNA binding protein, it was already known before us that from where it got its name, cold-inducible. So if you put cells in a cell culture at low temperature, human cells, like usually we culture human cells at 37, it's our body temperature, to like 32 or 33 Celsius, it start to accumulate CIRBP protein as a response to hypothermia. So mild hypothermia, so if we can somehow to decrease our body temperature, that's how is it by, I know, cold shower, uh, keeping ourselves in a cooler environment, or even doing calorie restriction, which has been shown to a little bit decrease our body temperature. This potentially can upregulate this protein in our body without even drugs intervention. Although the upregulation would be mild, it will not reach the level of bowhead whale CIRBP. Yeah. So some interventions searching for drugs which can mimic hypothermia and force the expression of this protein in humans, this is a way to go as a follow-up study. But as I said, some changes in the lifestyle can already be beneficial for us to slightly upregulate this protein in our body, and by this improve the DNA repair in our cells to help ourselves, because we are already protected with 5 hits, so which we need to tune a little bit some of our systems to make ourselves better. We are pretty long lived, right? And for our better, very much long lived and very cancer resistant, to be honest. And so we can tune our system a little bit. So I see that. I think the cool thing that this protein can be naturally upregulated besides the drug interventions. But that, of course, needs to be tested experimentally if it's sufficient enough to give some benefits.
We're long lived compared to other great apes, but it's not long enough for a lot of people. There's still such a huge market for how we can push it longer and push it longer. It's interesting that it's that tightly linked to environmental signals. And I'm curious if you think that this kind of potentially reflects a evolutionary niche for cold water environments, given that a number of species that hold lifespan records within a number of different clades, including outside of just mammals, are species that reside in these extremely cold Arctic conditions. Is that a fair observation?
It might be. We did not have access to the like some polar bears, for example, if what level of the protein they have. But our collaborators and we tested other marine mammals like dolphins, and it was pretty high there. Not to the level of bowhead whale, but it was pretty high. Interestingly, humpback whale have almost the same level of CIRBP as bowhead whale. The lifespan of this whale is less. I think, according to an age portal, it's around 95 years.
Mm-hmm.
So it might be, as you said, the adaptation for the cold environment, uh, natural adaptation that upregulate this protein to different levels. Because I understand that there is like a big vibe that this one protein, but of course we cannot explain everything in the bullhead well because of that one protein, right? It would be too magical to be true. There are other proteins which are differentially expressed even in DNA repair pathways, which might help and you know explain those phenotypes we found. It's just not a, we cannot in one single paper explain every single aspect and study every protein.
I really wanted to echo this because the real way that real science is done is to do what your team has done, which is to actually narrow down your focus and spend several years studying a specific pathway, a specific mechanism that plays a critical role in maintenance of an organism over this extended lifespan. But again, I'm going to echo what you just said. Just because this pathway has been discovered as playing a role in this extreme lifespan doesn't make it the unique feature that guarantees long lifespan, because it is a feature in other members of the clade, like in some of the other whales that live long-ish, but not extremely long, not multiple centuries. This is a similar observation also to some of the work that has been done on things like tumor suppressor copy number expansion in organisms related to elephants. And it appears that there is a similar phenomenon where this mechanism has evolved as a feature of the clade, but did not guarantee extreme lifespan in all members of the clade, but it laid the groundwork for the other adaptations that together made it possible for this evolutionary niche to produce extreme lifespan. And it does seem to be necessary for extreme lifespan, but it's not the magical one switch. And so many articles, including people who are already writing up responses in the informal media and academic space to your paper, are already making this mistake that you just pointed out and treating it like you guys found the one magical key that magically unlocked this hyper lifespan. So I appreciate greatly the fact that you kind of point out this is critical for lifespan. This isn't the magic pill for lifespan, though.
Exactly, exactly. So it would be surprising, right, if one single gene would be responsible for the entire explain aging and treat aging and forget about aging. Is it important that this protein is highly abundant in whale? 100%. Does it help? 100%. Is it the only factor which makes bowhead whale live longer? I doubt it.
Yeah. Yeah, we try to always, with every subsequent fascinating new lifespan related paper, we try to always remind people this is, if it's a mitochondria paper, we're like, mitochondrial aging is part of the picture. If it's DNA repair, this is part of the picture. If it's telomere maintenance, this is part of the picture. If it's tumor suppression, this is part of the picture. We're assembling this comprehensive understanding of all the different details. But how so to give the audience a realistic perspective on how the science really works, how many years of actual science went into the discoveries that got into this paper?
So let's see. I think together with the revisions, because we submitted in 2023 the paper to Nature, and there were 2 rounds of revisions. We had 5 reviewers, which will keep asking questions, and we made more and more experiments to make our findings more solid, to convince them. So from, I think initially, it is started when, even before I joined the lab in 2016. So it started around 2014 or 2015. So 2015, so almost 10 years from since we were able to establish whale fibroblast culture till you see paper online. It passed almost 10 years.
10 years, people. 10 years of
10
science goes
years.
into a paper like this.
Yeah,
Wow.
because someone got lucky and can publish Nature in few years. We spend a lot of time to find that niche, to find that protein, to find the phenotype which we can build on our discovery and our findings. So it was a very long journey, and that is why you saw there are so many co-authors on the paper from all over the world. We have collaborators from France, from UK, from many states in America, like all over the world. They were collaborators. There are people who were my students when I just came, my current student, and many people were involved to make this puzzle assembled to a way that it is deserved to be published.
Wow. Thank you so much for taking the time both to explain the discovery and to talk with us about the amount of work that goes into doing quality science like this. I hope this is a opportunity for anybody passing through and listening who is interested in longevity, who's interested in the evolution of extreme lifespans, or who's interested in pushing the frontiers of either human health span or even ultimately with the goal of extending human lifespan. We really appreciate both the hard work behind the scenes and the awesome job summarizing this in this beautiful paper. I hope to get more eyes on the paper and more people involved in the discussion of the actual complexity of these extreme lifespan cases. So thank you so much.
You're very welcome. You're very welcome.